All right, so want to welcome everyone to our spring guest lecture series, the Dwain L. Ford guest lecture series. And this spring we have, I think, about 15 different speakers and topics. And it's kind of interesting to look at them because we have speakers from, let's see, about 3 speakers from Michigan. We have a few from overseas, one from Canada, one from United Kingdom. Sometimes Texas tries to act as if it's a country by itself. We have one from Texas and so on. So we have actually 2 from the United Kingdom. And so we have a really nice lineup for you guys this semester. To lead us off in our spring series is someone who I'm just getting to know. He is at Eastern Michigan University, Professor Cory Emal. Is that correct, Emal?
Yep.
Yes. And so he received his PhD in chemical biology from University of Michigan, working with Professor Bill Roush. Is he retired now?
He's retired. He's doing something with a biotech in Boston.
Oh,
but he is retired from academia, yes.
Oh, okay. So at EMU, at Eastern Michigan University, which is where? Eastern Michigan University?
Is it Ypsilanti?
Ypsilanti.
Yeah.
Professor Emal works on bioactive molecules, and some of them were inhibitors to target inflammation-related conditions. However, in recent years, and he'll probably tell us this story, in recent years, he's focused on molecules important to fermentation. And he is co-founder and coordinator of the Fermentation Science program at Eastern Michigan University. A certified Cicerone, I have no idea what that is, but I guess we will learn. An award-winning home brewer and an enthusiastic collector of varietal honeys. I know what honey is, but I never put those 2 words, varietal honeys, together. So we'll learn a lot. I know I will. It's interesting for non-science people because fermentation is the basis, or is part of the process for cheese, for yogurt, for kimchi, for sauerkraut. So it's important, and I think everybody knows, for beer and wine. It's important in the food and beverage industry. So thank you so very much for accepting our invitation, and I look forward to your presentation.
No, thank you so much for inviting me. It's an honor. The seminar list you have is quite impressive. So I'm not sure how I fit in, but it's
You do.
it's an honor. It's an honor to be a part of this. So thank you for inviting me.
Thank you.
It truly is going to be fun, I think. So
All right.
let me go ahead and share this. And so the way that this talk is kind of structured is really just a very brief overview of fermentation. And we use it as a way to sort of talk a bit about how we got to this program, this relatively new program at Eastern that there aren't a lot of in the country right now. And how we get students involved in research as a part of this program that's a part of our chemistry department here. So hence the sort of unwieldy title, but it gave me leeway to talk about a lot of different things without knowing what I'd choose when I gave you the title. So just very generally, I want to talk about what fermentation is. And so fermentation is a process, right? So this is a process where, in a very sort of technical way we can think about it, is we've got microorganisms breaking down carbohydrates, typically sugars. They produce organic acids, or they produce alcohol and carbon dioxide, and they're doing this just as a function of living and reproducing. When we think about fermentation more generally as an act, as humans, we're think of it a bit more as just sort of using microorganisms to convert one set of starting materials into some sort of more desirable products. So humans have been fermenting things for thousands of years. When I teach our intro course and we spend a unit on hard cider, we spend a unit on sauerkraut, we spend a unit on cheese, it almost always starts with me saying how 4,000 years ago was probably the first times human accidentally made cheese, because that's when we started domesticating cows, and that's when we started having excess milk. And we started looking for ways to store these foods, to make them keep longer and to preserve them, and in some cases to make them more edible. And the great thing about fermentation is these organisms are everywhere. So most of the time, fermentation of a fruit or a vegetable or something can happen on its own, just by the natural microorganisms that live on the surface of those fruits and vegetables. And so really kind of what I tell my intro students what fermentation is it's just controlled rotting, right? When something rots, it goes bad when we think of it, but it's probably just fermenting. It just fermented in a way that wasn't pleasing to us. So when we ferment something to make foods and beverages, so if we're going to make pickles or kimchi, or pickled green beans or okra, or we're going to make cheese, or we're going to make beer or wine and cider, we're really just figuring out what are the carbohydrates that I can feed to these particular microorganisms and manipulate in a way so I get the product that I want, right? And so humans have been doing this accidentally for thousands of years, and once we started realizing what was happening, we've started to try and control it and to figure out how to make these processes work. And so things get a bit more complicated, right? It's one thing to just let grape juice ferment on its own, but it's another thing to harvest grains and process them in a way that we can make leavened bread, which we can do from the natural yeast and bacteria there. It's where sourdough bread, is just sort of this living culture of an enormous number of microorganisms. Cured meats, aged meats are typically fermented as well. That's something that can go wrong in ways that a lot of other fermentations can't. So I wouldn't recommend it be your first home fermentation if you get inspired by this talk, but that's certainly a way you can go. Most of the ethanol biofuels that are being produced are occurring by fermentation. There's just an enormous number of chemical starting materials and pharmaceutical building blocks and products that are produced by fermentation as well, by just getting these microorganisms to make what we wanted from these sort of plentiful starting materials that we give it. So with that sort of as a background, so why am I here and how did I get here? And so this is where, if you'll indulge me, I'll sort of tell you the winding story to why there is fermentation science at EMU and sort of what our program is, before I tell you why it is it's really a way for us to take a lot of our existing chemistry and biology courses, with a lot of focus on the lab courses, and integrate them in a way that brings together the science of fermentation and the context of fermentation to help make those particular courses, connect to things in the real world, but also to give our students who go through these courses, maybe a bit of a leg up to get into certain industries, because they now see the connections between how, my biochem lab can, help me be competitive for a job at a yogurt plant, or in an R&D facility, or in quality control at a winery, or these sorts of things, to bring these practical skills into that context. So we don't teach people to be brewers per se. We do, for example, we do brew beer on campus, and I teach a whole course on designing beer recipes and how we sort of leverage the underlying science to help us increase the quality of what we're trying to make and control the process. But we try to be agnostic and to sort of have our fermentation courses aim at a wide variety of potential careers. So there's a bunch of courses that sort of integrate with these chemistry and biology courses. The beverage production course I alluded to. We have a sensory analysis course, which is my favorite to teach. This is sort of the eating and drinking and smelling course, where we talk about all of these flavor active chemicals and how they're produced and how we can control their production, and how we think about setting up experiments with human beings as instruments to be able to, set up a tasting panel and to get real actionable data from that, these sorts of things. And so these courses, they're open to anybody in the university, whether they're in the program or not, just as long as they get the prereqs. We do see quite a few students who take the intro course as their science gen ed, and they get interested to take some of the advanced courses. And so that's been nice to see. So why does this exist? This 3-barrel brewing system that can make 100 gallons of beer, why is this on the 5th floor of our science building currently? So I started, when I went to college, I was going to be a high school science teacher. Wasn't really sure what at that point, probably physics or chemistry is kind of what I liked, but I went to University of Nebraska for undergraduate. I got involved in undergraduate research with my general chemistry professor who talked to me about doing some research projects over the summer. And so saying yes to that and getting involved in research really set me on a path I could never have predicted. It both sort of got me into the idea of experimental science as a way to better understand the world. And I also happened to meet my wife, my now wife, in that lab. So this has been very, very lucky for me on a couple of different pathways. And so, but I was going to be a high school teacher. And so when came my senior year, I went and did my student teaching at one of Lincoln's high schools. And I realized I didn't like teaching high school students. Like I was there to teach, not motivate. Nobody wanted to be there. So thought, well, let's go to grad school. Maybe I can get to the point where I can teach college students someday. And so I went to University of Michigan and got my degree in chemical biology, and I was basically an organic chemist. So I did synthetic organic chemistry for 5 years at Michigan, making essentially anti-malarial molecules. So again, it was sort of my interest was in how do I make organic molecules and how do they interact with biological systems. It was really sort of the idea behind medicine and what drove me to be interested in that. So eventually then I went and did a postdoc in Washington, DC, almost joined the Army. That was close call. I didn't because we started going to Iraq soon after that. So I missed that. But I came back to Eastern Michigan because I really liked Michigan. And they had an opening for an organic professor, and I applied, and luckily I got the job. And so for the first 10 years I was here at Eastern, again, I was still synthetic organic chemist doing medicinal chemistry, working on anti-inflammation compounds. And at a certain point during grad school, I was probably getting close to 30, I realized I liked beer. I didn't think I did most of my life, but at that point, but craft beer was starting to become a thing, and you could find beer that wasn't Budweiser or Coors. And it was just really fascinated by all these different flavors and everything. And so there was a local class, there's a get together where you could train to become a beer judge. And so I did that just because I wanted to learn the language. I wanted to be able to put terms to these things in the beverage that I was experiencing that I couldn't describe, but I really wanted to. I could understand the differences, but I couldn't figure out what it was. And as I learned more about that, I learned more about the science behind it, because all those different flavors are caused by different chemicals. And all those chemicals have a different source, whether it's from the ingredient, whether it's from an organism, it got there somehow. The guy Greg Wilmus in the office next to me, he kind of had the same, he was always kind of a foodie. And so he knew a lot about fermented foods. He's one of the people I know who can make aged sausage in their basement and not get people sick. And so we just kind of put our heads together and we decided we were going to do a chemistry of food class. And that eventually sort of spiraled out of control into us designing an entire major around it, which sort of coincided with this real boom in craft beer and craft beverages and foodie culture, and a lot of DIY home fermentations that people are doing, food, beverage, and otherwise. And so that's really kind of how we got here. There was no way that I could have planned my path to Eastern itself, from being an undergrad, let alone to helping start this program, which at the time, I think we were the fourth university in the country to have a fermentation science major specifically. So that's kind of my story of how I got here. I just kind of want to say, it's good to have a plan, but you don't need a plan. The plans change. It's okay. A lot of what you're preparing yourself to do is just to sort of recognize opportunity and take it when it shows up, right? And learn a lot of diverse skills. And so that's really the best thing I think you can get out of any education. So enough about me, though, and why I'm here. Let's sort of get to talking about a couple of fermentation specific projects. And so, again, just to reset ourselves with fermentation, we always kind of think of fermentation as sort of originating from glucose. It doesn't necessarily, but most organisms take whatever sugars they can metabolize and convert it into glucose and then metabolize from there. And what we see just from a basic scientific level is this glucose gets turned into pyruvate or pyruvic acid, and then broken down into acids or alcohol and carbon dioxide. But we get this really wide variety of different fermented products because we can get those carbohydrates from a lot of different sources in a lot of different forms. And we can throw all kinds of different organisms at them as well. And so just a few of them are listed here on this screen. Most of our alcohols are yeast fermentations. Saccharomyces is the most common one. Cheeses, we're typically throwing lactobacillus and lactococcus at it, but even just different types of cheese have a lot of different secondary organisms. Swiss cheese has Propionibacterium. Blue cheeses will have penicillium as the mold in it, and so on. We can do a lot of industrial products by fermentations as well. So Clostridium is, something you absolutely do not want in your food, but boy, it's a great way to make acetone industrially, right? If we just, again, feed it the right products and treat it the right way. and so on. Vinegars, soy sauces. Desmond and I were talking earlier about cocoa and chocolate. Coffee's got a fermentation step to it. There's all sorts of different organisms out there that have these important, or these products have these important fermentation steps to them. And so these two pathways here on the right are sort of the general types of fermentations we see. Down the center are these homolactic fermentations, are usually our acidic fermentations, where lactate is usually the major acid that's produced. Alcoholic fermentations usually produce ethanol and carbon dioxide. And these happen when the organism either doesn't have access to oxygen, can't use oxygen, so they're like an obligate anaerobe, or they just choose not to use oxygen. They choose to ferment. Saccharomyces yeast can do this often. If it's in a sugar-rich environment, it may not do this sort of oxidative respiration, which is much more efficient, but it's a lot more work. You have to build a lot more machinery to do that. And so a lot of times, if there's a lot of sugar and it can go to waste, yeast will just choose to do an alcoholic fermentation rather than respiration. Okay, so this is pretty simple, and at its root, this is fermentation. But the reason why wines taste different and why cheeses taste different and why we can control all these things by changing the organism is because organisms don't just do this, they do this. Right? So this is, an attempt to capture all the major biochemical pathways of yeast. There's all kinds of stuff going on here, not just respiration and metabolism. There's all these other pathways, and a lot of these compounds are flavor active or aroma active or have a pharmaceutical benefit or have some benefit to humans in some way. And if we know how to treat our microorganisms and feed them the right things, maybe we can ramp up a desirable pathway or shut off an undesirable pathway, depending on the context of what we're trying to make. And so what makes fermentation interesting to me is all these other things that are going on. Alcohol and acid, it's nice. That's maybe why mainly we're doing it, but it's not the interesting part. It's all these other chemicals. And so with all of this, we have a lot of opportunities to get students involved in research. And so from here on out, I'm going to describe a few of the research projects that we've done here at Eastern, one or two in a bit of detail, and some others just to give you a little flavor of the types of things that you can do that are connected to fermentation in some way, even if we're not specifically doing a fermentation in all of them. They're all connected to the process of making these fermented products. And so Desmond mentioned honey, and honey is something that I've become unfortunately infatuated with, which we'll sort of get into here. So honey is made by bees, right? So bees go out, they forage in blossoms, and they gather nectar, and they bring it back. As the nectar in a flower blossom is mainly sucrose and water. But there's a lot of other chemicals that come along for the ride. Basically, if two blossoms smell different, right? Those are different chemicals that are causing that difference in aroma. And those same chemicals can come back with that next nectar, with the bees back to the hive. And so when bees travel to different nectar sources, the honey that's produced from those sources can taste and smell and look differently, depending on the chemicals that are present in the blossoms where they're foraging. Okay. And so the conversion of that nectar to honey is fairly straightforward. The bees have something called a honey stomach that they bring the nectar, they store the nectar in to bring it back to the hive. And in that honey stomach are enzymes that break down sucrose, which is this disaccharide, into its constituent monosaccharides, glucose and fructose. And so honey is mainly a mixture of glucose and fructose. There is still some sucrose around. The bees, essentially vomit this out of their honey stomach into the honeycombs. And you've seen videos of bees waving around, beating their wings over the comb. What they're doing is they're evaporating the water out of the nectar, and they're concentrating the sugars. And they get the water content down to around 17%. And when you get it to that point, honey will keep indefinitely. It won't spoil, as long as you don't get it wet, because the water content is lower than bacteria. And so if any bacteria fall onto that honey, just the osmotic pressure will suck the water out of the bacteria. Nothing can really live there. But if where the bees are foraging, if it's enough of a monoculture that bees can go for miles, so this can sometimes be tricky. But with sort of modern agriculture, we have a lot of areas now that are essentially monocultures agriculturally. And so when the bees are predominantly visiting one type of plant, then this honey can take on a so-called varietal characteristic that's predictable. So buckwheat honeys are very dark honeys. In the East, they're often a little minty. Western buckwheat doesn't have the mint to it quite as much. It's a really robust tasting honey. Meadow foam has these enormous vanilla notes to them. There's this Italian honey. They have this tree they call the strawberry tree, which doesn't have strawberries, but the honey produced from these trees is very bitter in a way that you never thought honey could be. And so on. All of these different types of plants can provide these different sort of consistent, predictable types of aromas. And so mead makers, so people who make honey wine, essentially, so fermented honey, are really interested in particular in these varietal honeys, because when you ferment mead, when you make mead, you take honey and you dilute it in water, you put your yeast in, and the yeast consumes the sugars. And so the sugars go away, and what's left behind, in addition to the alcohol that the yeast produce, are all these flavor and aroma chemicals. And sometimes the yeast will modify those, sometimes they'll add their own to them. But if a mead maker wants to get a vanilla note in their final mead without using vanilla beans, well, they can use meadow foam honey, and they can get that character from it. So there's a real sort of culture of folks using these different types of honeys to get different flavor and aroma profiles. There's also a sort of medicinal side of things. There are certain types of honeys that are purported to have certain health benefits. Manuka honey from New Zealand is a big one. It's if you go down the aisle and there's one honey that's really expensive, odds are it's Manuka. And it's a bit of a problem because it only grows in New Zealand, the Manuka plants. And there is, I forget what the current numbers are, but there's something like 5 times more Manuka honey sold worldwide per year than is actually produced. So that's a problem because not all of that is Manuka honey. It can't be. And so as people get more interested in these varietal honeys from specific sources, counterfeiting and adulterating starts to become a problem. And so whether they're adulterated with other sugars like corn syrups and rice syrups, invert sugar, or whether they're prized varietals are cut with commodity varietals. So clover honey, avocado honey, alfalfa honey. These are all fine honeys. They taste like honey, but they tend to not have the special thing to them. So you can stretch your Manuka honey a lot further if you mix it with some clover, right? So our interest is in, can we develop a spectroscopic method to give ourselves any confidence whether a particular varietal honey is what it purports to be based on the chemical signatures of those particular honeys. And so I'll zip through this kind of quickly. This is, again, sort of meant to give you the flavor of this rather than actually explain NMR spectroscopy. But, you know, coming from my background as an organic chemist, NMR spectroscopy is something I've got a lot of experience with, and it's honestly, it's right across from my office. So it's my instrument of choice for a lot of reasons. But it's also, it can be very sensitive, and we can use it to look for specific chemical signatures that we might not be able to see in other analytical methods. And so what we want to be able to do is develop this method to see, is our honey adulterated, and is it the varietal that it says that it is? And we're working with a nonprofit industry group called the Mead Institute, which is actually based in Ferndale, Michigan, that is trying to develop these sorts of standards for the industry as well, because they're very sort of concerned about honey quality too. Okay. So the way we started with this is we sort of built a small database. From sort of reputable suppliers, we bought 5 different varietals that all 4 of these suppliers had. So we bought buckwheat, orange blossom, raspberry, clover, and sage honey from 4 different suppliers that we hoped were all getting them from different sources themselves. And when you look at honey by NMR, and so you just take some honey, dilute it in some deuterated water, and we get this spectrum. And I can show you any honey and it will pretty much look like this, because what we're seeing are the sugars here. The sugars are by far the major component of honey. And so they're what we see in an NMR spectrum. Because what we're looking at here is basically all of the hydrogens that are present in this chemical mixture and what their electromagnetic environment is at this snapshot in time. Okay. The good thing is that if you have different sugar sources, you know, the sugars look different, as you'd expect. So if you compare agave syrup to, say, Tupelo honey, which is all the way at the bottom here, you can see there's a lot of similarities, but there are quite a few distinct differences in those spectra. Corn syrup as well, very different from the other spectra we see here. Invert sugar, well, that's a bit of a problem. That, if you compare these two, the brown and the green, those are pretty similar. That's a tricky bit to tell apart just by a very broad-based look at the chemical signatures here. And if we look at different varietal honeys, we see the same thing. So these are those 5 different varietals. These are all from the same supplier. This is sage, raspberry, orange blossom, clover, and buckwheat honey. And again, this all looks the same. So how's this going to work? Well, we're looking at the big picture view. And we know that there must be chemicals in there that make them different, because we can taste the difference. We can see the difference between these honeys. And what you have to do is you have to zoom way in, right? You're not looking at the major compounds. You go way into the baseline and just keep zooming and zooming and zooming. And now you start to see these little flecks of compounds that show distinct differences between the varieties, you know. Humans are very sensitive. We're very adept at tasting and smelling different things. And so it doesn't take much for us to be able to get a different sense of two different varietal honeys. And if we sort of take a broader look, we can see there's a lot of sort of differences here and there, even between different buckwheat honeys. Agricultural products vary, by location to location. This buckwheat field, one of them might have been next to where they planted spearmint, 20 years ago, and the bees are still finding wild spearmint in that one location, and it gets in here. But we can see a lot of similarities. And it's our hope that we can use those similarities to help say, yes, this is a particular variety, and use the differences to discard the ones that don't fit the profile. And so we need fancy statistics to do this, because our puny human brains can't look at these little baseline flecks and put together a fingerprint. And so we found there were a couple of reports in the literature of people trying to do this with principal components analysis. And we decided instead to use a linear discriminant analysis. And I'm not going to get into the details of those, partly because that's why I collaborate with the statistician. Right. I can do the NMR, they can do the algorithms. But the choice of using this LDA is basically because we have knowledge. If we have a trusted set of honeys that we know came from a particular source, we can use that as a training set for the algorithm. And so that's part of the value that we have in working with the Mead Institute, is they're a conduit for us to the beekeepers and sort of the primary providers of the honey. So we can have sort of a chain of provenance with the samples that we use to ultimately build our database. And so, this kind of works. So if we take that test set of 20 honeys of 5 varieties from 4 suppliers, and we feed the data into the algorithm, and we say, okay, can you separate them? It does a really nice job. So each of these circles here are binning or finding similarities between all of the things we said were the same, and it can find a way to discriminate them from the others cleanly. And so that's if we feed the entire spectrum in. If we, early on, we were trying to make sure that we weren't getting a lot of noise into the spectrum, because baselines, they flutter, they're noisy. And so we used to discard the smallest 5% of those peaks. And when you do that, the analysis gets a lot worse. Now we've got these overlapping circles. Now the algorithm can't confidently tell raspberry apart from buckwheat, even when we tell it which one's raspberry and which one's buckwheat. And if you taste them side by side, they're nothing alike. You don't have to be a honey snob to be able to tell buckwheat from almost anything else. And so what that indicated to us is that it is those minor components that are important. That stuff in the baseline is helping discriminate. And sure enough, when we go back and we tell the algorithm to only look at the minor peaks, we get this version, which is exactly, I can't believe how exact this is. It is exactly the same results we get when we feed it everything. So when we give the algorithm the fire hose of data, it gives us the same results back as when we give it just the minor components. So this, this sort of gives us a lot of confidence for the future. And so this is kind of where this project is right now. Right now, we're working on sort of building our connections with beekeepers and getting as many trusted samples as we can into the database, so we can then start to challenge it with unknown samples and see how well it does that way. So this is sort of a project that we're building on, and is the most active one in my lab right now. Let's see. The other active project in my lab right now, and I'm going to zip through this a bit because, as I told you, Desmond, I can really talk sometimes. Is we're working with a brewery local to us to come up with a simpler way to measure the bitterness of beer using sort of a cheap UV spectrometer. Okay, so bitterness in beer comes from hops. So hops are this flowering vine during the boiling step of making beer, which is a fairly complicated process as far as fermentations go. Hops are added in there, and these alpha acids, these compounds here that are naturally present in beer, they isomerize into what are called isoalpha acids. And so it does this at temperatures over about 80 centigrade. It doesn't do it at low temperatures. And it's the isoalpha acids that are bitter. Non-boiled hops aren't particularly bitter, not when they're dissolved into beer. It takes heat to form these bitter compounds. And so brewers want to know how much of this compound they're producing. And it's a really tricky thing to measure directly because bitterness, humans sense bitterness, it in different ways. It's hugely dependent on our genetics. The best ways to measure IBUs are also the most expensive ways, and no brewery would ever have, an LCMS set up unless they're an enormous brewery. And so our friends over at Arbor, they wanted to know, is there a way that we can sort of do rough IBU testing so we can try to correlate it with what we're tasting while we're going through the process, just to get a rough sense. So if they need to change things midstream, they can basically taste how the process is going over the hours and days that it takes to make this beer. And the standard method that was used is really has some complicated equipment. It has solvents, it has a defoaming agent. They just want something simpler and faster. And humans can only taste to within a range of 5 to 10 IBU. And the most bitter beers only go up to about 100. So this doesn't need to be enormously precise for the purposes that they're using. And so there's a published method, right? And you need a shaker, you need a big centrifuge, centrifuge that can handle 50 ml centrifuge tubes, and you extract it into an isooctane layer, and you've got octanol and the special anti-foam thing. And you need the centrifuge because you always make these emulsions, and these emulsions are terrible. And so this is what we've been beating our head against, is trying to keep these emulsions from happening. And the short of it is we figured something out. So instead of shaking, we came up with this very easy method where you put the tubes in a lab rack and you put your notebook on top and you just do inversions, and you just count 200 inversions, and you can do 25 samples at a time without creating this emulsion, but getting good mixing. We're going to kind of work on reducing the amount of solvent needed next. But for the purposes here, we can get fairly consistent results. So we bought some commercial beer because Bell's has a lab. We figure they're going to be relatively consistent. And just going by their marketing, they report these two beers at being 60 and 20 IBUs respectively. And we get pretty close to that. We're off by maybe 10% or so on each of these, which, given how humans can't really taste 5 to 10 IBUs difference anyway, this is kind of close enough. So we're working with Arbor now to go in on some brew days and pull samples at different points in the process and see how the IBUs change over time during that process, so they can start to think about flavor as it relates to the process in front of them. Okay, a couple other quick one slide things, and then we wrap it up. Other stuff we've done just before the pandemic started. You know, this is just, you know, died because of the pandemic. We went up to EMU has a remote site called Fish Lake that's out about an hour north of Detroit. And so we went and we collected a lot of wild yeast samples there because we wanted to try and find something to brew with. We thought it'd be kind of cool to tie some wild organisms from Fish Lake there. And so we went out, you know, we found a bunch of stuff, and then, you know, March 2020 hit and all the stuff died in a cooler somewhere. But this is something we'll probably revive at some point. Is, you know, it's a great way to bring a lot of microbiology into a research project and find some useful organisms. Students who did that was about 15% of the soccer team at the time. So Aaliyah and Maggie and Aubrey worked on that for quite a bit. One of my colleagues, so we were just talking about bitterness and IBUs, he was using NMR to study the kinetics of how that isomerization occurs at different temperatures. And so by looking at the NMR spectrum of basically the before and the after, you can see the conversion of the alpha acids into the isoalpha acids. And because it's a relatively slow process at high temperature, you don't need complicated like stop flow technology or anything. You can just put a tube in 90-degree water for 10 minutes and then go take an NMR and then keep doing that every 10 minutes. So you can you can do some interesting kinetics experiments that way. Bunch of other stuff we've done. Here's some of our students out presenting things. Mel did some stuff on feeding different microorganisms, different carbohydrates to see how that changed their production of antibiotics. Olivia was doing some wild organism capture from ciders. Justin did some sanitation method stuff. Kim is who is working on the IBU things. We've got students in classes doing stuff as class projects. And here they all are. So none of this happens without student researchers. You know, there's not enough hours in my day to do it myself. So the sort of training students is really one of the joys of the job. We've got some support from great places, and that's where you can find us.
Okay.
And I ran over, Desmond, but I really tried.
Okay.
I'm sorry.
You did better than a lot. So we appreciate your presentation. Thank you so very much. I learned a lot and gave me some ideas as well. I mean, I never thought about taking an NMR of honey. That gave me some other ideas. But anyway, are there questions? Let's open up for questions.
Yep.
Ian, you could ask a question. Ian? Well, I don't know. Okay, Ian's mic is broken, so he wants to know, what is your favorite beer?
Oh, what is my favorite beer? That's I'll give you two, because
Okay.
I can't. There's not just one. My desert island beer is a beer called Weinstephaner Hefeweizen. It's a German wheat beer that has certain bits of its genome intact compared to most other brewing yeast, and it produces notes of banana and cloves. Isoamyl acetate and 4-vinyl guaiacol are the two compounds that it makes that really give it that profile, and it's something special. The other one is a Belgian beer called St. Bernardus Abbot 12. And so again, Belgian yeast just do all kinds of wild things. And this smells like figs and lilies, and I can't even tell you what else, but it's new every time I have it.
Wow. Well, I learned a lot also about it's, you know, simple chemistry, but the formation of honey. Like I never really thought about what was involved, never looked it up, but taking sucrose and then essentially dehydrating it down to 17% water.
Yep.
That's an interesting process that I didn't know about.
Yeah. There's a lot of wild stuff going on with honey so I said that honey was just sort of antibiotic because of the low water content, but what is also going on in honey is also converting some of the glucose into gluconic acid. And so it basically breaks open glucose and forms a carboxylic acid. And there's another enzyme that does that. That process also produces a little bit of hydrogen peroxide along the way. So what's happening as honey is being formed is it's becoming more acidic, which makes it better preserved. But then it's also got this layer of peroxide protection on top of it. And so this idea of if you've got an open wound and you've got nothing else to, you know, to rub on it, to put some honey on it, it's not entirely wrong to say that could work. There's a reasonable amount of peroxide in honey.
Oh, wow.
Yeah.
Wow. So the increase in the acidity also, isn't that going to make it even a better antiseptic?
Yep.
Yeah?
Yeah.
Yeah.
Yep.
Okay.
That's one of the key things, I think, if you're doing fermentations at home, particularly if you're pickling vegetables and doing things like that, you don't want to taste your fermentations too early because you want to give them time to acidify. Most of the enteric bacteria that give us food poisoning, they can't survive below a pH of about 4 to be safe. 4.5, is okay. But sort of letting it acidify down past there helps kill off most of the baddies. And then the rest of what's around is often the sort of things that's just found in our gut, right? So these wild communities really can sort of give you sort of better overall health when consumed the proper amounts, of course.
Yeah, so another idea that came to me here is, okay, what's the most acidic honey? Is that something that's known?
I'm sure it is, and I don't know off the top of my head what it would be. Yeah, I never thought about it.
Yeah, because I'm thinking one of the experiments that we are doing and have been doing is using lime and lemons and apples and stuff, juice, which are acidic, to do organic reactions. To, for example, make azo dyes. And it's been successful. So my my thought is like, if we get an acidic honey, maybe that could also be a catalyst.
Right. No, that's an interesting. I've never thought of using honey as a catalyst in that way. I like that idea.
I know. Thanks for putting it in my head.
Hey, that was my goal with this. If one person has one good idea, then I did my job.
All right, that sounds good. Other questions? So we had a question on, okay, go ahead.
I'll ask one, yeah. A great talk, always interesting to see, and hear about natural products, but I may have missed this. Do you guys actually, you got some big reactors there, do you guys bottle that and sell it as like a fundraiser for the lab? Or?
Yeah, we're not licensed, so we can't do anything other than give it away. So we in what's kind of a sad story is that that got installed. That picture was taken again in March of 2020. And we basically brewed our first batch of beer on it, and we all got sent home 5 days later
Yeah.
for a long time. And so
Yeah.
we're just getting to the point now where we're starting to have on-campus events. We're starting to do things again that we might be able to use this at. So we're going to fire it up again this semester and make a big batch and see what kind of takers we have. We sort of designed the space with the idea that it could be licensed eventually, but we need somebody to run that who is not me.
Yeah.
Right.
Yeah.
Yeah.
Are you guys also looking at any fermenting or other, just maybe using it as a to ferment to make some other molecules, or as a resource for other, targeted fermentation development?
Yeah, we really never really thought about using that system for anything other than its intended purpose, but that doesn't mean that there couldn't be something.
Sure.
We had considered there is one or two local places to us that have talked to us about using it to. It only becomes illegal for us to sell it once it becomes alcohol, but before we add yeast, we could still sell the pre-fermented wort to people. And so there are a couple of people that have asked about using our system, and then they carry it away in sanitized totes. We haven't really gone down that route yet, because they haven't gotten quite serious enough. But there is a way that we could use it, open it up to the community in that way.
Yeah. Interesting.
And there is, a history now of fermentation being used to make either intermediates towards pharmaceuticals or pharmaceuticals directly. So that's another, yeah.
In one of those just sort of most serendipitous moments, it was about a year ago, I guess now, is I got a call from somebody I knew at a small biotech company in Novi, who wanted to know if I had any students who are interested in internship, because they'd just gotten a small fermenter in specifically to make some new products in their company that they wanted to sell. And as it turned out, someone who just graduated he'd gone to school in Mexico for his engineering degree, and then he'd come to us to pick up some fermentation classes. And I was like, yes, here is Pablo's number. And so, Pablo went and worked for them for 6 or 8 months to get that set up and get him off and running before he went on to the next thing. But that's, again, it's just getting into the fermentation side of things has really got me connected to a part of, to a part of industry in certain communities in Southeast Michigan I never would have been part of before. And I've been able to use that to do some interesting things for the program, but also make these connections for students, because now I know, I overlap between circles of people who don't know each other, and I can get them connected, even if I can't do the thing either of them, want to do. I know who to get them to talk to.
So that opens up opportunities, employment opportunities for your students.
Yep.
So you said that your fermentation science program is one of a few.
I've lost track now. It's kind of slowed down. There was a little bit of a spike. Depending on how you count, at least a year ago, there were maybe 10, 15 places you could get a 4-year degree in something vaguely like fermentation science. Like some places, like Western Michigan they call those sustainable craft brewing or something like that. So I'd count that, right? So things like that. But there's a lot of certificate programs. There's a lot of places that have, developed online classes and stuff, but in terms of, we're still one of the 5 or 6 places that focuses on the science as the guiding principle with it all, because partly that's because that's what our background, our strength is in the science. I don't have a background as a professional brewer, so I don't want to be the person teaching people to be professional brewers, because that's not my training. But I can teach science, I can teach these skills, and you can take those skills and then go work anywhere that has a lab. It doesn't have to be in fermentation, right? It's learning how to streak out cells on an agar plate is a skill you can take anywhere. You don't have to take it to Dannon.
Do you guys have a biotechnology program? Because I'm assuming that in a biotechnology program, they would cover some aspects of fermentation.
Yeah, we don't have anything specifically like that here. It's something that would be a nice overlap with our College of Engineering, and obviously with our program in chemistry and biology, and we have a hotel restaurant management program as well. But yeah, there hasn't been one thing yet to coalesce like that would be kind of neatly tie a lot of these things together.
Right. Okay, if there are no more questions or comments, this was a very good presentation to lead off. I mean, something as simple as, quote unquote, simple as honey, I haven't given a whole lot of thought to how exactly it was made and all the different things that you brought up and stuff. It was a learning experience to me, hopefully for all of us. So I want to thank you very much. And I know offline, you and I are going to be talking.
Sounds good.
All right.
Happy chat. Thank you all. I appreciate it.
Okay. Take care. Have a good evening.
Thank you.
You too.
Okay. Bye-bye.